Thank you for joining us today. Today we're going to cover the Annexin 5 assay on Celigo. You know, there's a lot of people doing the apoptosis assay in such communities like the immuno-oncology, and I figured this was kind of relevant to bring back into our webinar session if this is something that interests you. Okay, so first we're going to give you an introduction of what the apoptosis and the Annexin assay is, and then I'll tell you how I prepared my plate with the staining of the cells in the plate today. And then we're going to cover how to acquire and analyze the images on Celigo with the PS externalization application. It's very easy, 3-channel application. And then I'm going to show you another more advanced method where we can acquire 4 channels with bright field. This application is a little bit more advanced. It's customizable and it uses gating. So you have an easy version and then you're going to have a little bit more complex version. All right, so the apoptosis is a natural process for cellular self-destruction. It's a part of the routine cell turnover and tissue homeostasis. It's prevalent in epithelial cells, erythrocytes, and other cell types genetically programmed to have their limited lifespan. It's important for embryogenesis, maintenance of immune tolerance, and development of the nervous system. It can be induced either by a stimulus, such as irradiation or toxic drugs, or by the removal of a repressor agent. The cells disintegrate into membrane-bound particles that are then eliminated by phagocytosis. When there are problems with this regulation of apoptosis, it's thought to be linked to many cancers, degenerative diseases. So this is why people are interested in studying it. So when you're looking at cells that are going through apoptosis, you can generally find bright field morphologies that are changing, such as the plasma membrane blebbing, chromatin condensation, nuclear fragmentation, and formation of apoptotic bodies. That's just from visually looking at it from the bright field view. Then you can use some more biochemical criteria to identify the cells going through apoptosis, such as the mitochondrial membrane permeability changes, caspase-dependent activation, DNA damage, and then cytochrome C, and then what we're going to look at is the phosphatidylserine membrane exposure. So now this is going to be using the Annexin 5 binding externalization assay on the membrane. So the Annexin 5 is a member of the Annexin 5 family of intracellular proteins that bind to phosphatidylserine in calcium-dependent manner. So in healthy cells, this PS molecule is normally found on the inside of the cells in the plasma membrane, but during apoptosis, it actually translocates to the external leaflet of the membrane. Using this, we can actually come in with a fluorochrome-labeled Annexin 5 to specifically target and identify those cells on the surface of the apoptotic cells, as shown in the diagram here on the right. So you can see a healthy cell with the PS on the inside of the membrane, and then as it goes to apoptosis, the red molecule goes to the outside, and then we can bind it with the Annexin 5 FTSE-labeled fluorochrome. So here's an example image where you have Hooke-stained nuclei and then Annexin 5 labeled apoptotic cells. So this is Annexin 5 binding alone cannot differentiate between apoptotic and late-stage apoptotic or necrotic cells. So then we use propidium iodide to come in and label the dead cells or late apoptotic cells. They will only come into the membrane-compromised cells, and then if you have a double-stained apoptosis Annexin 5 and PI stained, you can identify the late apoptotic cells versus the early apoptosis, which is Annexin 5 only cells. Okay, so for the plate that we're going to show you today, the reagents that we used are the Annexin 5 FTSE, the Annexin 5 binding buffer, propidium iodide staining solution, Hooke staining solution, and then the apoptosis inducing agents. Then you'll also need some pipettes, some tips, the tubes for mixing the reagents, and then the Celigo imaging instrument. So in general, the assay protocol is plating cells In a 96 well plate, approximately 5 to 10,000 cells per well. Let them adhere overnight. The next day you come in and add the apoptotic agent. In this experiment we used staurosporin, and a second drug on a second half of the plate was alpha-TOS. This will help identify the positive and negative controls. You'll incubate for X number of hours. This can be 6 hours, 24 hours, 96 hours. And then you're going to create a master mix of the stains, and then you're going to remove the well liquid and add the master mix to the wells. Let this sit for 30 minutes, and then you come in and add more binding buffer, and then you image on Celigo. If you want more information on the lab portion of this, you can look on our website. So now how to acquire the images on Celigo. So in the Start tab, we're just going to load a 96 well plate. And in this case, it's a Greiner 655090 plate. And then you'll give a plate ID. In this case, we'll just label it Apoptosis with Annexin 5, Propidium iodide, and Hoechst. And then you'll just click Load Plate. The stage will open up, and you'll put the plate on the stage, and then click OK. The stage will go back inside the system. Now we'll move over to the Scan tab, and the first thing we'll need to do is select the application PS externalization. It's about 2/3 the way down on this list. It is a 3-channel application, so we'll be collecting or imaging 3 channels: the PSE, the dead channel, and the total channel. So once you've selected this application, the channels will be displayed up here, and we'll first change to the total channel, where we're going to set up the Hoechst nuclei. I prefer to set this up because then I know all the cells have content, and we can set up the focus channel registration on this channel. So first we'll move to a well on the plate map, and then just click Snap, because we know that the Hoechst is blue, and I just want to see where we are for focus. It's a little bit dim, so let's click 80,000 and then click Snap. So now the cells they're dim and they're a little out of focus. So let's first register the focus, get it in focus. So we'll click Focus Setup and then Register Auto. The system will actually move up and down in Z, kind of like you're looking on a microscope, and get the cells in focus. And then when it's finished, it will have registered the hardware focus mechanism to now, at this point, focus at that position for every well across the whole plate. Okay, so you can see the cells are in focus and it's registered. So we'll close the dialog box, and we can zoom in to take a peek at our cells. These are the Hoechst stain nuclei. That looks good. Now let's switch over to the green channel, the PSE channel, and we're going to change the illumination to green. And then adjust the exposure. So for this assay with the kit that I used, it's about 60,000 exposure works. And then you click Snap to see where you are. They're a little out of focus, so let's use the word Find Focus. Now this will actually move the Z up and down and find the green channel focus position, and it's going to produce a number just above where we clicked that tells us how many microns above or below the focus registration we did for the blue channel. And so, I mean, we need to image the green channel 23 microns above the blue channel. So we'll click Set Offset. So the system will now acquire all green channels 23 microns above the blue channel, which is our Hoechst stain cells. That's where these cells are in focus. That number is populated up here in the focus offset box. Now we can do the same for the dead channel. So let's switch over to the dead. And we'll change the illumination to red and adjust the exposure. For this PI staining of 60,000 will work. and then click Snap to see where we are. There's a few dead cells in the image, so let's just click Find Focus. This will again study the picture or the image set in multiple Z's and figure out what is the best focal plane for the dead PI cells to be in focus relative to the blue hook staining. When it's finished, it'll produce a number just above where we clicked. It says -11 microns. So at this point, we'll click the word Set Offset, and it'll populate that number in the dialog box up top. It says Focus Offset. So the red image will be collected 11 microns lower than the blue channel. So now we've set up all 3 channels for imaging, and we can select the wells on the plate map for acquisition. And then click Start Scan. The system will now start collecting the images for those wells according to the parameters we just set up. And we've moved over to the Analyze tab. And now we can start looking at any of the wells that have turned green on the plate map because those have been saved to the hard drive. And while the rest of the plate's being acquired in images, we can start setting up our analysis parameters. Okay, so here we have a well with all 3 channels on. We can turn on and off any channel at any time or display them all at once. To start setting up the graphic overlay, which is what is identified in each of the images, we're going to start with the total channel. So just turn on the image and the graphic overlay for total. And now we'll identify the hook stain. The parameters on the left-hand side are are default to kind of put you in the ballpark of where you should start. Let's first change the channel over to total so we can update any parameters. And also I use the auto analyze. Make sure that's checked so that anytime you adjust the parameters, it automatically will update the image graphic overlay. So I just adjusted the intensity threshold so it'll separate some of these close nuclei. By increasing it, it's basically detecting less around each of the nuclei. And then now we can move over to the image display and graphic overlay settings for the dead channel. And so there isn't a whole lot in this well, so we might want to move over to a treated well, something that has a lot more dead cells in it. You can see the blue overlays. There's not many of them. Let's click on A2. And there we have a lot more dead cells. So the blue overlay is identifying anything that's stained with propydium iodide. And it's doing a pretty good job with the default settings. We might want to remove some of these very small specks that are not necessarily dead cells. So we'll filter these out. So the change over to the dead feature type and increase the minimum cell area requirement from 10 to 30. And you can see the small items have been removed. And then I just toggle on and off the graphic overlay to properly see that I've got picked out the cells. And then we can move over to the PS or Annexin 5 stained cells. So we'll turn on the image and graphic overlay for this green channel. And we have a lot of big bright Annexin 5 positive cells. Then we have a lot of extracellular debris that's been left behind from the cells maybe that have detached or moved around a bit. So we'll exclude those by increasing the minimum cell area requirement. So from 10 to 50. That seems to be doing a pretty good job. Okay, so that's pretty much it. There isn't a whole lot to play around with. I think the intensity threshold is pretty good at 3. And now we can select the wells that we want to analyze, which is all of the ones we're imaging, and then click Start Analysis. This moves us over to the Results tab. And the image acquisition is continuing on for the rest of the wells, and the analysis parameters are now being applied to all the images that have been saved in the hard drive. So you can see that we have the thumbnail image for the PS channel. We can just double click on a well and the high resolution image is displayed. You can zoom in and see how our graphic overlay is identifying the PS positive cells. And there we have some double stained. We have PI and PS, or Annexin 5 stained cells. And then we have identified all of our cells. Now the thing is, the yellow outline is for the nuclei, the purple is for the dead, and the blue is for the Annexin 5. Or, sorry, I have that backwards. Purple is Annexin 5. Now if there is green objects that are not touching a nuclei, they're going to be excluded from the analysis and considered debris. So you don't have to worry about those extra small little cellular particles. If it's not touching the nuclei, it's going to be excluded. So you can see we've properly scored all of our cells for Annexin 5 and dead. Now in the results section, you can see on the upper left-hand side, as we move around, the top portion is presenting a percent of PS dead or PS corrected. The PS corrected is basically cells that are scored for PI, the PS corrected is Annexin only. The PS positive is actually for Annexin 5 and PI. So that would be your late apoptotic cells, and the corrected would be your early apoptotic cells. So it's doing a pretty good job. I think that we can go back to the results tab and let the software finish analyzing the rest of the plate. And so you can see on the left-hand side, you can change the display of what's on the thumbnails by clicking the bars on the left. So currently we're looking at the Annexin 5 only. There's a lot of different information available. So you have the percentages, the counts, and the intensities. And this section below is just the well that you've currently selected on the thumbnail. Okay, so the software is finished analyzing all of the wells. And so now we can actually export the well level data into a CSV file, which can then be looked at in Excel for graphing. Okay, so here is the plate level information, and below that we have the percentages for the data of those wells. This is showing the Annexin 5 and PI double stained, PI only, and then the Annexin 5 only. Now I want to show you another way of acquiring and analyzing the images in a little bit more advanced method. This would actually include a bright field image, so you can look more at the morphology of the cells going through apoptosis. So we're going to look for the expression analysis application, and we're looking for target 123 mask. This is a 4-channel application, and this will allow us to collect a bright field image in addition to the 3 fluorescent channels. This application is customizable, so you just click this little icon up here and you can change the channel names to be something more relevant to your experiment. So for this case, we'll do green, red, bright field, and blue. The channel is the image, the feature is the analysis features of the graphic overlay, and then the gated subpopulations are what we're going to identify in the gate tab. Those are our classifications. So we'll use the hooks to identify all of the cells, and then we'll look in that same area in the green and red channel and ask, do we have Annexin 5 staining, do we have double staining, or do we have just PI staining or no staining at all for those cells? This will become more clear when we get to the gating tab. Okay, so here we've rechanged the names to be more in line with what we're going to image. So let's start with the blue channel. This is going to be our hook stain, and we'll set the illumination up for blue and change the exposure to 80,000, and then click Snap. You can see the cells are there, but they're a little out of focus. So we'll set the focus registration using Focus Setup and then Register Auto. This is the same procedure that we did in the previous one for the blue channel. The system will move up and down and find the right focal plane for the blue hook stain and register the hardware focus mechanism. Now all the wells will be collected using that Z position for the blue channel, and we'll set up the other channels with their illumination and offsets to the blue channel. So let's start with the green channel. Change the illumination to green. Adjust the exposure. For this application, 60,000 works for the Annexin 5 staining. And then click Snap to see how it looks. So the cells are there, they're a little out of focus, so we'll just use the Find Focus button, which allows, the system will find the right focal plane for the green channel, and then it'll produce a number that tells us how many microns higher or below, above or below the blue channel. So in this case, it's 28 microns above the blue channel. So we'll tell the system this is the offset we want to collect the green channel is in relative to blue. And then we'll change over to the red channel and adjust the illumination to red. Set the exposure to 60,000 and then click Snap. So again, we have a few cells there. That's enough for the system to find the right focus. So let's use that Find Focus button. So I'm using the system more to help me find the right focus. If you prefer to do it manually, you can use the up and down arrows. But once you find the right focal plane, click the Set Offset button, because that will be the offset value that will be used to acquire the red channel. So in this case, it's -16. So that's -16 microns from the blue channel. The red image is in focus. And then the third channel will be bright field. So we'll just use the AutoCalc button. This was designed to help give you the best background image for the bright field. So it says about 4,000 in this well. Those cells look pretty good. I don't feel like I need to set any offset. I like the focal plane that they are going to be imaged in. So this is pretty good. So we don't need to change the focal offsets for this. Then you just click Start Scan. And now we've moved over to the Analyze tab. We'll click on a well, and you can see now we have 4 channels all displayed. So this is one of the advantages of having this 4-channel application. You can get a bright field image to observe morphological changes. We talked about the blebbing of the cells. So that is another feature that you can see here with the bright field image in addition to your fluorescent images. And also the advantage to this is you only have to set up the graphic overlay on the Analyze tab for the blue nuclei stain. So here the default settings 4 and 10 are pretty good. You might want to increase the intensity threshold to help separate touching nuclei, but you can also use separate touching objects. That helps break up some cells that maybe have just gone through division and haven't quite moved away from each other. The other feature I'd like to have you use is the dilation of the radius. Because our staining might be just beyond the nuclear area, you can increase it by a pixel, so you'll see all of the yellow overlays increase by 2 pixels in diameter. So there we go, or radius. Okay, so that's all you have to set up for the Analysis tab. At this point, we can move over to the Gating tab. So just oh, one more thing. Sorry, before we go there. We would like to observe the pixel intensity of the background for the red and green channels. So just turning on the red and green images alone, we can start to run our mouse over those images, and the background values are displayed down below. So we'll use this information to help us set up the plots in the Gating tab. So there's a little bit of background with the Annexin 5, so we'll just note that in our in the gating that'll help us set up where to put the gates. So now we can click on the Gate tab. Now we'll select a well, and the whole well is visible at this point. It's stitched together and all images are on, except for Brightfield. And the graphic overlay comes on automatically. So you can turn that on and off just to check out your cells. So you can see our apoptotic cells. Now there's the Brightfield image. That's pretty cool. Okay, so let's start making a plot. So using the green plus sign, I want to make a scatter plot of the red mean intensity versus green mean intensity. So this dialog box pops up. We're going to use the source population total, which is all the blue graphic overlays on the image. And then we're going to use a scatter plot. And then the plot parameters for the x-axis, we're going to use red mean intensity. So this is all the PI positive cells. We're going to plot all of the objects according to their mean pixel intensity in the red channel. And for the y-axis, we're going to use green mean intensity. So all the objects identified from the nuclear hook staining will look in the green and red channels and plot the objects according to their red and green pixel intensity. So you can see here on the scatter plot, we have the camera range is from 0 to 255. So all the objects should fit somewhere in that range. So really intense Annexin 5 staining will be really high. And then PI is on this x-axis, so we can see the camera range again is from 0 to 255. Okay, so let's go to a control well that doesn't have a whole lot of Annexin 5 staining. It's just a few cells that have those stains. All right, now I'd like to create some gates to actually score the ones that are showing up with green and red, or just red only. So how do we do that? Well, first I want to zoom in on this scatter plot. We're kind of looking spread out, but I want to focus the visualization into the small portion of the graph. So let's change the parameters of the axes with this little button here. And I'm going to adjust the red mean intensity, so we're really focused in on where the objects are on the scatter plot. So let's change it from 0 to, I think, let's do about 30. So then we can really hone in on those objects there. And then also let's do the same for the green mean intensity. So let's change this so we can focus in on the cells that are just transitioning from non-stained Annexin 5 into staining. So let's go from 30 to 60. So I really want to look at the objects close to the range where they switch over from not stained to stained. And that's what we see here displayed. So we have a huge cluster of cells that are not stained, and then it starts to get a little bit more stained, and there's a few cells that are at the top portion of the graph between 50 and 60. This is where we need to focus our gate. You remember when I was talking about looking at the pixel intensity of the background? So we're going to use that information to help us place this quadrant gate. So clicking on this little icon here and then clicking on the screen will actually divide it into 4 quadrants. But to place it, I'd like to actually use that background information. So we had red at 10 and green at 49. So just adding about 4 pixels to, or intensities to that, we can place the scatter plot to 14 and 53. And now the green mean intensity will basically have scored positive cells in the upper left because that's above green background. And we can change the color to actually outline every cell in green for that upper left-hand quadrant. And then we'll checkbox that class to be Annexin 5 positive, PI negative. That's the first one after total. So that's how that class will be reported in the Results tab. And then the upper right-hand corner is scoring cells positive for green mean intensity, or Annexin 5 cells, and PI. So we'll want to change this one color to about red, and then check box Annexin 5 positive, PI positive cells. This would be your late apoptotic cells. And then the right-hand, we'll start with the lower left population. This is our healthy cells, negative. Currently, it's scored colored green. We want to change that to something that's maybe blue would be a good color because it's not green or red. And then we'll checkbox this one as Annexin 5 PI negative. So those are our healthy cells. And then the last one is the lower right-hand quadrant, which is currently colored as purple. Let's change that to a red color. That's over here on the right-hand side of the quadrant with red mean intensity. And change that color to red. And checkbox the third one, which is the fourth one, Annexin 5 negative, PI positive. Okay, so now we've properly classified those 4 quadrants and scored them to their mean intensity for both the green and red channels. And you can see the graphic overlay has also changed the colors. So we have scored Annexin 5 only in green, yellow is double stained, and red is PI only stained. So this is a control well. We'll want to look at a treated well that has cells in both channels. So let's change the well location over to a treated well, something that will score for a lot of Annexin 5. So here you have a lot of cells only with Annexin 5, so you can see the green quadrant increased in objects. And the graphic overlay is also showcasing green-only cells. If you want to see what your data look like for this particular well, you can click the word results here, and it gives a bird's eye view of what is going on in this well for data. And this is the data displayed in results. If you want to make any adjustments at this point, you can click on that square in the middle of the quadrant and move your gate around. Once you're happy with the gates, you can click Start Analysis, and it'll apply this gate to all of the wells. And you can see the data displayed on the top is Annexin 5 only, so early apoptosis cells in a percentage. And so turning on the heat map can color code the wells to showcase your high scored wells versus your low scored. And you can change the data display to have your different classes showing up in the percentages. Okay, so there we have showed how you can present 4 classes. And again, I want to double click on a well and show the reason why this is an advantage is now you can also see your bright field image and look at bright field morphology changes before you even get Annexin 5 staining. So one thing I noted in studying this Annexin 5 assay is that sometimes the cells will just round up but look healthy. They won't have any Annexin 5 staining. So you can see we have some rounded up cells that didn't stain with Annexin 5. So that's an earlier indicator versus the Annexin 5 staining. So it's just one more advantage. If you're looking into studying apoptosis, you can include a bright field morphological feature in your image analysis. So. Okay, and then turning on the class graphic overlay, you can just observe which cells got scored for any one of the 4 classes that we've defined in the gating tab. And it's done a pretty good job. Okay, and then here's a control well or less drug. And the percentages are displayed at the left-hand column along with the counts and the pixel intensities for those channels. Now we can go back to reports, or excuse me, the plate level, and then export this data into a CSV file, which can be observed into the Excel program. Okay, and then let's see how the data is displayed in Excel. Again, you have your plate information on the top half, and then the classes that we defined in the gating tab are displayed below. So first is the percentages. You can see the Annexin 5 only, the double stained, and then the PI only. And then below that should be the negative or healthy cells. And then below that are the counts for the classes that we defined. So those are counts that were picked up from the gating tab. And then below the counts, you'll see the total, which is the hook-stained cells. So you can get, that's where your percentages denominator is coming from. Okay, so in summary, we've covered quite a bit in this application. We've showed you the apoptosis and Annexin 5 assay the method how to stain your cells, how to acquire your samples easily on Celigo, and then a more complex, advanced method, but incorporating a bright field image in your acquisition. So at this point, I know you're going to have a lot of questions, so let's open up the forum for questions. We covered a lot today.
Thank you, Sarah. We do have some questions. All right, so your first question today is, what is the difference between focus, setup, register auto, and find focus?
That's a good question. I get a lot from people. So the first thing you want to do is the focus setup. That is actually engaging how am I going to have the system find the right focus from one well to the next. And so hardware focus is actually a mechanism that once you or the system defines, because we're using the auto registration, it's finding the right focus for that setup well. Then it applies these hardware focus beams on the bottom of the plate. And then when you move from the next to the next well, just the beams are actually engaged. So it's assuming that the plate is very flat and the focus should be the very same position. But the thing is, the fluorescent channels for the others, so we focused on blue as our focus registration channel, but the green and red channel actually fluoresce in slightly different wavelengths, and they need to be imaged in slightly different heights from one channel to the next. So if we registered in blue, the green and red were registered in offsets that were microns different from the blue channel. So you need to tell the system to actually do that other fluorescent channel imaging at slightly different heights than you did at the blue channel. So by setting the offset, that's actually programming the system to acquire those other channels in slightly different heights than they did the blue channel, so that those channels will be in focus. I know it's complex. Did that answer the question?
We'll find out.
Okay.
Sanjay, if you have additional follow-up questions, feel free to type them in and we can certainly address those. All right, so your next question, Sarah, is asking, does setting different offsets for each channel increase the time of image acquisition?
The Z position changes for those different focal offsets for the different channels isn't that much. You wouldn't even notice it. I would say where you lose time or where you get slowed down is when you have increased exposure time. So I designed this assay to be at a concentration that puts you in an exposure time that is acceptable with good signal and low background, but low exposure. So we were using 60,000 and 80,000 exposures. That's a respectable exposure. If you go up above that, you're going to slow your acquisition down.
Okay, great. All right. So the next question, is saying that they noticed you did not scan the wells on the edge of the plate, both the top and the bottom on the rows and the columns. And is this due to an instrument limitation?
It's not. I just chose to minimize my data set on this plate. You can image the whole plate. The system will acquire any plate that fits in an SBS format. So you can do 1536, 96, 384. I just, in time for this teaching lesson, I chose to do less number of wells, and the data set was a little bit quicker. If you have, let me just comment though, if you have assays that are going to require more than 24-hour incubation, you might want to exclude the outer wells because they tend to have evaporation around the outer, the most outer ring of the plate, and the growth patterns of that will show some differences. So I typically will exclude those if you're going to have a long assay, more than 2 days.
Okay. Sarah, just to follow up on that question, haven't we also found that some plates, depending on the brand, the manufacturer, the type of plates, that the actual production or manufacture of the plates makes there some irregularities on those plates, which makes the focus not consistent throughout the entire plate?
This is also true. I know that we've had we've evaluated some of these plates to kind of learn that the outer edge of the plate is also got thickness issues. So it's a thin film bottom, and the thin film is actually more, let's see, less consistent on the outer wells. And so if you're using the hardware focus mechanism, the thickness is going to be a little bit, well, let me stop this. Stop. The thickness on the outer ring of the wells is going to be a little bit different, and that's another reason you might want to exclude the outer edges of the plate. Alternatively, if you don't want to have that less number of wells, I might recommend you move into a 384 well plate. I know it's a little bit daunting to think about pipetting into that. And another potential option is to do a 96 well half area, because those tend to have less issues. We're going to look into that type of plate to see how well it performs in the outer ring compared to the rest of the plate.
Okay. Great. The next question is the analysis of green and red stain limited to within the blue overlay that is first defined?
So yes. So the idea of the mask, which is identifying the blue nuclei, is to identify all of the cells. And so I used the nuclear area to identify all the cells, and then I used the dilation radius of 2 pixels, which is 2 microns, beyond what it found to expand it a little bit beyond the nuclear area. So you're basically trying to score just a little bit of the cytoplasm. You don't need to capture the whole cell. You just want to have a little bit into the cytoplasm.
All right. We have an unrelated question. Can this instrument be used to image larger cells in colonies or spheroids? I guess the question is the overlay analysis, is it going to work correctly?
Are you looking to identify a single cell inside of a sphere? That is a little bit tricky. A 3D format, as you go up and down in Z, your fluorescent signature is diminished. So I don't know about the spheroid detecting single cells, but we can detect a spheroid in bright field and give you pixel intensity of the spheres in the green, red, or blue channel. For flat 2D colonies, we can detect the cells. The overlay analysis may not work. For flat colonies, it should work just fine. We also have other applications that will define the colony as a whole and give you pixels within there, if it's green, red, or blue, of the whole entire colony. If you want to email us, we can probably help that last question out, maybe get a little bit more specific. I'm not certain I answered it.
Okay. All right. So, Sanjay, if you want, I can put you in touch with Sarah at the conclusion of today's presentation, and she can kind of get into a little bit more detail with you on that if you'd like. Perfect. I'm not seeing right now any other questions come through. I'm going to give people just another second in case something comes to mind. Also, if you're watching this as a recording and you have any questions that might not have been asked or answered today, you can always reach out to support@nexilom.com, or you can give us a call at 978-327-5340. You can follow the prompts from the automated little voice to reach our technical support department. And one of our technical specialists and application scientists will be able to assist you with any questions you may have.
Sarah,
Again,
I want to.
Yeah,
don't forget to mention next month I want to cover another kind of assay for apoptosis. It's using caspase 3.7. So a lot of people are interested in this kind of assay. So we've developed this on the Celigo, and I'd like to share that with the people next month.
Okay.